We continue our study of asexual reproduction in protozoans. In this video, our focus is on this organism which is called as a slipper animalcule because it looks like a slipper. This is a paramecium. Paramecium undergoes transverse binary fission, which is what we are going to discuss in this video. Let's start with the structure of the paramecium. Similar to Euglena, Paramecium 2 has a proteinaceous pellicle. It has an oral surface which contains the oral groove, the cytostome, and the cytopharynx. It has two nuclei. One, the macronucleus, which is polyploid. This nucleus controls the cell's daily activities and metabolic activities such as growth, digestion, osmoregulation, etc. Therefore, this nucleus is also called as the somatic nucleus of the paramecium. The other one is the micronucleus, which is diploid, and this is involved in genetic recombination and sexual reproduction. This is the germline nucleus. The paramecium also has two contractile vacuole, the anterior contractile vacuole and the posterior contractile vacuole. Since paramecium lives in freshwater environments, water constantly enters into the cell. These two contractile vacuoles help in collecting all these excess water and then pumping it out of the cell. Under the pellicle, we have the infraciliary system, which is responsible for the coordinated movement of the cilia. The infraciliary system hosts the trichosis below them. These are primarily defensive structures, and when they are triggered, they discharge these sticky filaments against their predators. The sticky filaments can also help them to adhere to surfaces sometimes. Above the infraciliary system, we have the cilia. They are going to extend from within the body of the paramecium to the outside. Cilia is useful for locomotion. Transverse binary fission occurs only when the conditions are favorable. During this period, the paramecium starts feeding until it reaches its maximum size, and then it stops. As the cell is preparing to divide, we notice that the oral groove in the parental cell starts to disappear. Then we start seeing these changes in the nucleus. So, starting with our micronucleus, it starts to divide by mitosis. Mitosis ensures that the daughter micronuclei receives DNA that are genetically identical to that of the parent. The macronucleus, on the other hand, it divides by amitosis. So here there is no organization of the chromosomes and division. There is simple elongation of the entire material and then they just divide. So this might lead to unequal distribution of chromosomes between the two daughter cells. So we have learned that asexual reproduction usually leads to clones. So in this case, do you still think the progeny would end up as clones? Actually, the answer is yes. But to know why, watch out for our video on sexual reproduction in protozoans. Once the nuclei are in their end stages of division, there are these transverse grooves that appear in the cell. And they appear almost in the middle of the cell and they deepen to give two daughter cells. We also see that the oral groove that has disappeared at the start of the cell division is slowly making a comeback. So the two daughter cells are the anterior Proter and the posterior Opisthe. Between the two daughter cells, there are few structures that they inherit from the parent, but few of the structures they have to create new on their own. In case of Proter, it receives anterior contractile vacuole, cytopharynx, and cytostome from the parent itself. It has to create its own oral groove as well as the posterior contractile vacuole. But in case of Opisthe, it receives only the posterior contractile vacuole. It creates a new oral groove, a cytopharynx, a cytostome, as well as the anterior contractile vacuole. So this is how the parental structures are distributed between the two daughter cells. Since the direction of the fission or the plane of fission is perpendicular to the body of the paramecium, so the paramecium's body is longitudinal, top-down axis, and the division happens across it, right? This sort of fission is called as homothetogenic fission. On a previous video where we discussed cilia, we learnt about the kinetes. These two run longitudinally along the direction of the paramecium's body. The plane of fission here is perpendicular to the kinetes too. Hence, it's also called as perkinetal fission. Under favorable conditions, the fission takes about 2 hours to produce these two cells, which are considered as the first generation of daughter cells. And if the conditions continue to be favorable, the paramecium can produce 4 generations of daughter individuals in a single day. We have been discussing about the binary fission that occurs during favorable conditions, right? We look at asexual reproduction during unfavorable conditions, which is multiple fission. In multiple fission, multiple offsprings are produced in a single division of the parental cell. During unfavorable conditions, the organism actually doesn't divide. It's only at the end of it, the organism is able to divide. So during such unfavorable conditions, the cell prepares itself for division, and finally, when it is actually able to divide, it releases multiple offsprings. In this manner, it is able to increase its chances of survival and reproduction. For example, the amoeba, which usually divides by binary fission, undergoes sporulation during multiple fission. It is also seen in Plasmodium, which is a malarial parasite. Plasmodium has an elaborate life cycle that is shared between the human and the mosquito. And multiple fission occurs many times within this life cycle. So we have schizogony, male gametogony, as well as sporogony happening during these cycles. We'll learn more about this when we learn about the life cycle of Plasmodium. So let's look at how sporulation happens in amoeba. One of the first thing amoeba does during unfavorable condition is that it retracts its pseudopodia and forms a cell like this. Then it secretes a hard covering around the cell called as a cyst. The cyst forms a thick protective multi-layered coating around the cell. Inside the cyst, the nucleus of the amoeba undergoes multiple mitotic divisions without any cytokinesis. This forms many nuclei. After nuclear division, the cytoplasm also divides. They divide into as many bits as the nuclei, and they start to encircle these daughter nuclei. The result is the formation of many smaller individuals from a single parent. These are called as pseudopodiospores in case of amoeba. You also notice that the cyst is slowly dissolving. On the return of favorable conditions, the cyst bursts open and it releases the daughter cells into the environment.